So we want to listen to the next speaker in the person of Professor Fernando Buarque from the Computer Engineering program in the School of Engineering of University of Pernambuco in Brazil. Professor Fernando is a Brazilian, but with several international affiliations. He's a vast man in his field of artificial intelligence. And this afternoon, he will be speaking to us on curbing the data volume and combinatorial complexity on gene regulatory networks with swarm intelligence. Let's listen to Prof. Welcome on board. Thank you.
Hi, Bolandi. Thank you very much. It's a real pleasure to be with you all here. And also, after a magnificent talk from Carlos, I feel humble, but let's see what I can add. So let me share the screen here. Can you see my screen now?
Yes, we can.
But the presentation, the slides?
Yes, we can see the slide.
All right, so let me start with the title. When you, Bolandi, invited me, I paid attention to the topic of the workshop. And then I thought of what could I add in terms of give an example of how computational intelligence could be effective on problems that matter. So I don't know how familiar the audience is with gene regulatory networks and also with swarm intelligence. But the idea here is to show how this very complex problem can be solved in a non-exact manner by means of using swarm intelligence. Okay, so let me move on. So the agenda is the following. Basically, we have three parts. I'll try to be very compact in terms of packing three heavy topics like GRN, swarm intelligence, especially the physical search, which is the algorithm we created and later on was modified and be part of PALLAS. So that's the latter part of my presentation, is to explain how this mixed FSS was seminal for the solution of GRN in a much faster way. Okay, but before that, I would like to make some acknowledgments here to the very fine invitation I received from you in Akuri, from the TWOS Young Affiliate Network. I would like to thank the current fellowship I hold from FACEP, which is the state foundation here in Brazil, and Alexander von Humboldt. Also my universities, as Bolandi mentioned, I have some affiliations with Exeter, Johannesburg, Münster, and Texas A&M. But I'm based in Pernambuco. That's my main university. Well, I come from Recife, and the reason I mention this is because I already told that to Bolandi. I am open to collaborations. And I would like to show just for a second what Recife is about. So we are by the sea line, and we have beautiful beaches. Recife is called the Venice of the North, and the only problem is that it's so beautiful that some sharks now are very fond of gathering here. So the beach on the right-hand side is full of sharks. But luckily enough, Recife in Portuguese means reef. So we have a very good understanding. The sharks become on one side of the reefs, and then the swimmers on the other one. Anyway, another interesting thing about Recife and Akure in Nigeria is that although now we are very apart, almost 5,000 kilometers, in the past, in the Jurassic period, we were together. So our dinosaurs, they probably could walk from Akure to Recife in a couple of hours. So that's just to show that we have other links than we imagine. Before the beginning as well, I would like to just mention very briefly that in our group, we tend to use meta-heuristics, we use computational semiotics, and we are using those techniques to solve problems in medicine, in health, in logistics, which is health, by the way, is one of the topics of your workshop. Our research group is called CERD. We have lots of connections, and Nigeria could be the next link we add to this network. So, but let me talk very briefly about gene regulatory networks. I don't know how familiar you are, but if you take problems such as the airplanes taking off and landing in one particular moment. So this animation is just a period of 6 hours, and every dot is a plane taking off in the US. Of course, this is an animated and a fast-forward version. But the reason I'm showing you this is that we live in a very complex world these days, and it's for the lay person, complexity may only lay on the real world and the visible one. However, the problem that Carlos mentioned about the curse of dimensionality is really real and hard to come, because as the elements of the system grow, it becomes more difficult for you to make predictions or optimizations, etc. So the direct impact of that is that computational costs should be tamed. This gentleman here, it's called Hermes Trismegisto, was a philosopher in the Middle Ages. He had a very interesting thought that what happens in the macro world happens in the micro world. So with this, I very quickly would like to draw complexity from the visible world, like the universe, to brains and cells, okay? And this brings us to another implication, that scalability also must be considered in this level of granularity. I don't know if you are familiar with a cell, but this is a nice illustration of Encyclopedia Britannica. A cell that seems a very tiny element is really complex. This is just for you to see the organs inside a cell. They are very many, and they have various signaling mechanisms. Of course, it is nice as scientists for us to understand these processes, but the reason for that is because we may help to improve health of people if we know how these processes work. Obviously, the outside of the nucleus is the inner part of the cell, is basically to support the function of the nucleus. Okay, so here you have a perspective of the nucleus. All right, so you have all the chromosomes inside the nucleus, etc. But the main point of the nucleus is not necessarily to store the chromosome. Inside the nucleus, there is another structure called the nucleolus, which is responsible for the synthesis of proteins and peptides and many other compounds. So basically, within the nucleus of a cell, there is a structure that is very much works like a factory, producing enzymes and all sorts of things on demand. Of course, the recipes, if you like, are stored in the chromosomes. But the problem is we don't know what triggers these processes, because all sorts of things emerge from here. For example, you can create an enzyme to fight something, for example, COVID or a virus or whatever. But the network that creates this is a sequence of events in time and space that it's hard to track. And if we master this, for example, we could have in the future cure for cancer and big things like that. All right, so if you would like to see more, I separated here some links for you to follow. But that's the first part. The second part is just to talk a little bit about swarm intelligence. Carlos mentioned a particular set of families regarding problems that have multiple objectives. Here, I will be more humble and I will just deal with one objective, which is the inference of the network that controls the process. But for that, I have to explain, for those of you, I don't know how familiar you are with these techniques, and I will use the FSS family, fish school search, which is the algorithm we created in our lab about 10 years ago, that has some features that are very useful for the problem I described. So that's the first paper. It came out in SMC 2008, okay? And basically FSS was invented not only to be another member of the family, but to solve a high dimensional problems in high dimensional space with particular things in mind. But here, just for you to understand, we have a set of fish, we call them fish, which is processing units that perform individually local search, while the school, I mean the collection of the fish, aggregates this social information and solves the global optimization problems. Unique to FSS, different from all the other techniques that we are used to, is there is an automatic control for exploration and exploitation, which is nice because once you deploy FSS in a dynamic environment, it controls when it should do breadth first search or depth first search in an automatic fashion. And then the centerpiece of that is a concept that we use called the barycenter. Yes, the fish, every fish here has a concept called the weight of the fish. And then you have a barycenter, which is a global variable that concentrates the weights of all the fish. Basically, if a fish is moving towards a nice area of the search space, it puts on weight, and therefore it drifts a little bit the barycenter towards the goal. In this case, in a single objective optimization fashion. So in short, FSS has features such as it can follow, it reacts as a group, it wanders around, so it's good for search, and it diverges. So for example, as I'm going to show, we can split the swarm automatically as well. So swimming here is a connotation for a local search together with global search. The success here is measured in two ways. So if the fish is large, so this particular fish or solution for the problem is good. And if the radius of the swarm is small, it's better. It means that the whole swarm converged to a point which is interesting. And as I said, the barycenter in that particular case would be closer to the optima. I mean optima because, as I mentioned, we can split the swarm. So, and then there are a few other details. We don't have time for this, so forgive me. I will be very quick here, because otherwise I would not reach the important part of the presentation. So this presentation will be shared, so you can read about the terms and all the details. But basically, what I can tell you is that together, the swarm can really find solutions in very large search space, okay? So we have basically two operators, the feeding operator and the swimming operator. The swimming operator is how the fish move, and the feeding operation is how the fish individually they learn. This is the sequence we used in the vanilla version of the algorithm, and this is the semantics. So, for example, individual movement is the local search. Then the fish learns if it moves towards a nice area of the search space, so it puts on weight. If it doesn't, it reduces the weight. Then we have the collective instinct movement, which is just a social glue. So the fish, they have to be somehow attached to other fish, otherwise it would be individual search. And then there is another concept, which is collective volitive, and this particular one is responsible for that phenomenon I mentioned to you of expansion or reduction in radius of the search, so that we can have movements in the search space that are coherent with the search object. There are many stop conditions possible. If you would like to see the formula of all that, you can go to this link. We don't have time for that. But here is an illustration of the three components. So imagine if you are looking for this red dot, and then you have the fishes in the initial positions, like the green triangles. When we use the operator, the individual movement, the fish try to search around the position. And then, as you can see, there is no coherence here. Then we apply this collective instinct, which is the glue, which is the average of the movements of the previous movement, so that there is another vectorial component trying to put the fish coherently towards an area, okay? And then the third component is that the one I mentioned to you that moves the fish inwards or outwards. Okay, so this is the illustration of the formulas I have not shown, but you can check. But let's see how this works in a simple function, like the sphere function. From A to B, C, D, and up to H, you can see how the swarm moves towards the objective, which in this case is in the center. So initially you see a radius, and then it slowly not only reduces the radius, but also moves towards the target. Let's see now a comparison between FSS and PSO, specifically on that matter of not having the flocking behavior. What I'm going to show you is just the movement of the fish towards the areas, in this case red. As you can see, it's very good PSO, but if you do not restart it, it will be working as a pack when the dynamic system happens. Let's see how FSS works because of that operator I mentioned to you. So it can converge, but after the search space moves, it spans very quickly and regroups in another point. So this is nice because it's automatic, okay? But then we have other families like the weight version, which is another family, another member of the family. Here, the difference is that the swarm can split based on the weight. So, for example, if the fish which is leading the pack becomes small, then there is a breakage between the links, and they try to find better leaders. I will just show the illustration of that using the equal peaks function. That's the top view. With time, as you can see, the subswarms, they split and they move towards every peak. In this case, it's not multi-objective, here is multimodal, which is a nice characteristics, by the way. Anyway, but let's see a comparison between another earlier version of the FSS, which use that particular PSO fashion thing. So it was able to converge, but with time, it becomes a blob. So we dropped the D version, and then that's what I presented you. We have the W version. When the background changes, the swarms, they regroup and split in a suitable manner. Well, this is the tools we are going to use. Okay, so there are lots of highlights. I'm concerned with time. I will just skip all that. There is a chapter in a book covering the 10 first years of FSS. But basically, I presented you two versions of that, the W, the vanilla, of course, the D. But today, I'm going to show you a very wise use of this mixed version. That's a joke. I mean, perhaps in the future, we'll have the receive version with sharks in it to just incorporate penalization function. Well, there is a website for that you can follow. But let's talk in the latter part of the presentation about PALLAS, which is this approach for the problem I mentioned in the beginning. So this is the base paper for this part of the presentation. So PALLAS means penalized maximum likelihood and particle swarm for inference of gene regulatory networks for time series data. So basically, what you have here is you have a time series, okay, showing the expression of genes, and then we will explore that for inferring what could be the networks that would generate that signal. It's a very complex problem because it's a combinatorial one. So the ideas behind PALLAS are that the ones I mentioned to you, it's an inference method, okay? But it used what we call POBDS, which is partially observable Boolean dynamic systems, which is the collaboration I have with Ulysses. You may look at this particular paper where you have the description of POBDS. And I will talk in the next slide why POBDS was an improvement in the state of the art, but why it need help and why it need help of FSS. So PALLAS, in all, is something able to scale to networks of realistic size. So this is key. With POBDS, you can solve reasonably well the inference of small gene regulatory networks, but it does not scale well. So basically, the idea here is to take this meta-heuristic and put this to work towards a better scalability of what we have. And for that, we have to create a variation of FSS, which is a very clever concoction of a PhD student shared between myself and Ulysses that mixed continuous and discrete optimization. Perhaps Carlos could tell me if there are many other ones in this regard. So basically, just to revisit very quickly, GRN, gene regulatory networks. So it has been modeled mathematically by several means. Linear ones, Bayesian, neural nets, differential equation, information theory, with different extent. Many of those, they have some limitations. Scalability is the most important one. But one particular one, which is the Boolean network, has a very strong assumption, which is you assume you have to have prior knowledge. I mean, you have to have complete observation of the gene activation and inactivation, which is not plausible. Then comes POBDS, which is the work I mentioned by Ulysses. In this particular case, you do not need to use complete observation, but, which is very nice, but it's not scalable. So that's the story, okay? So then it comes PALLAS. In a lay people definition, PALLAS is a sophisticated state space method that can detect edge directionality and activation and inhibition of genes, of course, without a prior knowledge, in addition of being capable of working directly with gene expression, without the need of course, the binarization I mentioned to you. So for that, FSS, the mixed version, was instrumental. The first appearance of mixedFSS was in MLSP 2018. This is the paper. And the key, I think this is the most important line of the whole presentation, okay? It does efficient simultaneous maximization of a penalized likelihood over discrete space and maximization also of the inference of the networks that of the parameters observed in continuous space. So it's a very, I think it's a very nice way to put together in the same optimization problem, discrete and continuous space with all the features I mentioned before. Just for you, why this matters? Because this kind of problem scales up very quickly. So this is the way you can calculate the scalability of this. It scales with the number of genes. For example, if you have a network with 4 genes, you have almost 700 million combinations of networks. If this goes to 10 genes, which is very small in biological processes, the number of combinations is 10 to the power of 50, which is completely unfeasible. By the way, this accounts for edge parameter, this accounts for regulation bias parameter, just for you to understand why this scales up. Now I have some examples to wrap it up. This experiment is in that paper I mentioned to you, is based on synthetic data. So it's a network with 10 genes, but in a very, and then you can see that there are inhibitions and then there are excitations, etc. And then you have PALLAS compared with other competitors. As you can see, we are the best one in terms of sensitivity, in terms of the identification of the network. Okay, specificity, we are almost as good as the other two. We lose a lot for Banjo, but pay attention that Banjo does not have the sensitivity we do. So we claim that we are now a very good contender because we can account for the 3 aspects in this inference process. Now, 2 more experiments very quickly. For example, the E. coli SOS DNA repair system. This is real data. And what you see in the red links is inferred by PALLAS. So we did not know that before. By the way, the data was retrieved from here, and you can follow in the paper the details of the simulation. Here is another real data simulation. Again, red arrows are linked to that. And what I can add to wrap it up is that with this, we are able to really go up and try to map networks that perhaps are linked to problems like cancer, as I mentioned in the beginning. So what is next? As I mentioned, POBDS were able to solve small nets, but if you have large nets, the computation time was in the order of months. If you would like to do that, it's infeasible. So when we used our approach, we reduce from months to PALLAS, I mean, to months to hours, okay? But even so, some networks, they will take weeks. Therefore, the next step, we are already working with that. So the first collaboration is with Texas A&M. So Ulysses, Yukun are there. But then we use the second collaboration, which is with the University of Münster. There is a collaborator there that worked with parallel computing. So we are parallelizing PALLAS, okay? So I'm very excited with that because we can now move from hours to minutes. Okay, so that's for me. Sorry, I tried to do my best. Oseun. Oh, by the way, Bolandi, there are lots of people around that can understand Yoruba here.
Yes, yes, you are right, Prof. Thank you for ending with that thankful note, Oseun. We are really grateful for that nice presentation, very exploratory and comprehensive about gene regulatory networks, how we can generate networks from genes and map them to problems. I know this will be very insightful for those who are